Okay, thank you for the kind introduction. And I'm going to switch. I'm going to move over here. Sorry. As I'm right-handed, it's easier, and I can look at you. So I'm also going to switch because we heard a lot about vaccines already. And I'm going to now explain on how to produce them. And so that's a little switch there. But before that, I was asked to present a little bit on our institute. So I come from Magdeburg. This is not very well known. We say it's in the middle of Germany. There is a river, the River Elbe, which just goes along Magdeburg. And it's also sometimes with strong rains, flooding. And the constructors of our institute decided it's a great spot to be next to the river. So they placed our institute next to the river. However, so it's over here somewhere, when the flooding comes, it comes. And we had to evacuate our, somehow the pointer is not, I'm too far away probably. Okay, so this was 2013. We had a flooding, and within 2 days, the level was really dramatically increasing, and we had to evacuate the cellars of our building and to put electricity with generators to save everything that we had, our freezers and so on. So it's quite an issue for us. Every time it starts raining, we get nervous. What else is known for the area we are? It's a water crossing, actually. And so you have over the River Elbe a canal that moves boats over. Another famous building in Magdeburg is this. It's called the Green Citadel. It's green because there's a lot of trees around the building, and it's one of the last buildings that Hundertwasser was known for. Good, so this is about Magdeburg. Oh yeah, I forgot. Very famous. We have a football club. It's playing in the 3rd division, and whole Magdeburg is crazy about it. So whenever they play, the whole city is moving. Last year we were playing 2nd division, but this year it's again 3rd division. So our institute has four directors, so 4 departments. And we are very special again in the Max Planck Society. We're an institute that is allowed to focus on engineering sciences, and with that we are allowed to do applied sciences instead of fundamental sciences. So we are putting a bridge. We have fundamental research and also applied. And this is why I'm also going to talk about the vaccine production. We have groups that cover systems biology, model reduction, with Professor Benner, crystallization, this is Professor Seidel-Morgenstern, fuel cells, Professor Sundmacher, and my boss on the bioprocess engineering, Udo Reichel. We also have some group leaders, so these are independent groups that focus also a lot on systems biology, developing mathematical and software tools. We have molecular simulations and designs for binding structures. Very new in our house last year, electrochemical energy conversion with Tanja Vidakovic-Koch, and a Max Planck fellow, Professor Antulas, who's in our house since 1 year. The group that I am in, the bioprocess engineering group, then has several teams. My team is upstream processing. We have molecular biology, bioprocess analytics, where we also have a little spin-off company, Glycera, very strongly in glycomics, the mathematical modeling team with my boss, then also very recent synthetic glycobiotechnology and metaproteomics. We're very close to the university campus in Magdeburg, and my boss has a chair as well at the university and is running a master course on biosystems engineering. So we have also there some lab space. Okay. Now switch to the topic on vaccine production, and mainly there what we have been working on since several years now is to try to intensify this production. So what is process intensification? So this is very easy. You go from a big machine to a very small machine. you have high concentrations of fish, or I don't know, how do you want to do that?
Because you need egg on this side. Yeah.
Yeah, so I always say next. Okay, good. So yeah, so you can do high amounts of fish, or a lot of people on one motorcycle, or a lot of cargo boxes onto these big transport boats. Next, with animal cell culture, this was first done with the animals, then we were able to get the cells into small scale cultivation vessels. Then if you go to production, we were at these big production vessels, and now we have been also switching to go into small production vessels. And this is what I'm going to talk about also in the next. So, but before that, I'm going to explain on what viruses we are working on. So mainly we have started with influenza virus. And influenza virus is a negative strand RNA virus, which is 100 nanometers large. And if you think on how much viruses are produced per cell, if you do really well, if you have a great cell to produce that, it's 20,000 virions per cell that you get out. We are also working with modified Vaccinia ankara. Yeah, we're switching the pointer, which is nice. I guess every speaker today needs to suffer through these. Okay. Now I just need to go back one more. Okay, good. So I come back to modified Vaccinia ankara virus, which is a double-strand DNA virus, and which is can be used also as a vector. So you could use it to produce malaria or HIV, but it's also very interesting now for gene therapy approaches. This virus is 300 to 400 nanometers large, and as you need it to still be able to infect the cells, you need infectious particles. And here the outcome, if you do well, is 10 to 100 infectious virions per cell, so much lower than for influenza. If you take yellow fever virus and Zika virus, or the whole family of flaviviruses, you're much less good. This is again a positive strand RNA virus, which with a small size of 40 to 50 nanometers. And here again, you need the infectious virions, and there you are, if you do well, you're at 10 virions per cell. So how do you produce these viruses? So a classical production process is using chicken eggs. But nowadays we also know you can use animal cells to do that. And first animal cells that you were able to produce in large scale were cells that were growing in adherent mode. So you needed always a surface to grow on. If you do that, you have at the beginning a tissue flask where you have a flat surface where you grow the cells. If you go larger, you use roller bottles. These look like normal bottles, and the inside of the surface is just used for the cells to grow on. And if you want to go into a bioreactor, you need small beads to have the cells to grow on. And what you can do, I didn't press. So what you can do is you can grow these cells on microcarriers, and you have about 2 million cells per ml in this bioreactor. And before you want, how do you produce these virions now is you first grow the cells to a maximum cell density, then you infect all the cells with virus, and you wait till the virus has replicated in your cells, and that's when you then harvest your viruses. So your product, your final product, is the virus, either active, infectious, or inactive as a total amount. But we have been able now also to change these cells to grow in suspension. And this is much easier to scale up because you don't need to have the cells grow on a surface. When the surface is full, then you cannot grow on. Cells that grow in suspension, you can just go on and scale up. So this is easier. And with the development of new media, you can now grow these cells up to easily 10 million cells per ml. So compared to the 2 million cells per ml for adherent cells, this was already a factor of 5 that you could increase your productivity. So the idea is to grow your cells to maximum cell density of 10 million and then infect the cells again. However, very easily at that point, it was seen that actually the virus was not really replicating well. So you lost your virus productivity per cell. So at this high concentration of 10 million cells that you were now able to do, somehow your productivity was lost. So you needed to think of something else. And we have two approaches that we follow on this. So one is to still increase the concentration, but to keep the cell-specific yield. But there you need to know a little bit on the infection strategy, and I will explain on that. And the next is you go for an extended virus production. So you go for a continuous process. And for that, you would need 2 bioreactors, and then we'll explain to you that. However, here come into play defective interfering particles, which I will also explain. Sorry, fine. Next, please. So how do you do that to get high concentrations of cells? So there are different ways of doing that, and what you actually need is, if you grow suspension cells, you need a device that retains your cells. So there are different ways of doing that. One is a spin filter, another is a hydrocyclone, or a disc centrifuge, or an inclined settler. And all of them are plugged to a bioreactor. The cells are put into, or moved through the tubes into that perfusion system. The liquid is removed while the cells are retained inside the equipment, and then the cells are pushed back into the bioreactor. All these perfusion systems are working with sedimentation. Next, then there's another perfusion system which is called an acoustic filter. Here an acoustic wave is keeping the cells in sort of a line interference frame picture. And so it is also able, with this acoustic wave, to sort of keep the cells in place in that little compartment. And you can move the liquid away and can then remove the medium and then remove the wave and then push back the cells into your bioreactor. Again, this is a system that you have no binding to anything. It's just keeping the cells in it. Next. And the last way what you can use is membrane-based cell retention. And there are 2 ways. One is a tangential flow filtration, which comes also from downstream processing, so from purification systems. And you push the cells through the membrane and back in a loop into the bioreactor. Alternatively, you have an alternating tangential flow system. Here you push the cells into a hollow fiber and push them back into the bioreactor. And this you do with a diaphragma membrane that you use for pushing the cells back and in. This is the hollow fiber module, which has membranes in there that have defined cutoffs, and through here you're then taking off the liquid and you're pushing back the cells. So all of these systems are allowing you to keep the cells, remove the liquid, and then push the cells back into the bioreactor, and you do this stepwise, and with that you increase the cell concentration. Next, please. So this is also very well known for animal cell culture processes to produce recombinant proteins. And what you then have, if you look at the concentration over the time in your bioreactor, your cells are growing to a certain concentration, and your product is produced in recombinant proteins. Well, for recombinant proteins, what they do is actually they do a cell bleeding here to keep the concentration at 40 million cells per ml, because they realize that glycoproteins that are very important for example, monoclonal antibodies, they are losing their structure if you go too high concentration. So what they do is they keep this system at 40 million cells per ml. If we do this now for viruses, we have our cells that grow to a certain maximum concentration. We infect with the virus, and now as the virus is replicating inside the cells, the virus is my product. However, it also kills my cells. So my cell numbers are going down instead of having them at a constant level. So what for me is important is to use this system really to get the highest cell concentration that I get, then do my virus production, and then harvest my virus. Okay, next. So this is what we did first try with influenza in our bioreactors that we have. These are 1 liter bioreactors, and we use this hollow fiber module with the ATF, so moving the cells into the hollow fiber and back again. And what you can see here, so you will see those slides often enough. You will have the viable cell density or the cell concentration over time here. And this line always indicates when we do the virus infection. And here you see the virus produced. So these are different ways of showing this virus titer. This is the total virus titer, and this is the infectious virus titer for influenza. So what you see is, if you just do this without such a perfusion system, you reach up to 5 million cells per ml, you infect, and then your cells go die and produce on the same time the virus. If we use a perfusion system, we can go up to 50 million cells per ml and then produce the virus. And you already see that we have, of course, been able to increase the virus productivity, but how about cell-specific productivity? Please, next. So the question is, do I keep the productivity per cell? And that's what we could show. So in this case, we were able to produce in batch as well as with this perfusion system 1,200 virions per cell. Next. So we were really satisfied. We could show that 50 million cells per ml was okay. So now what about MVA? Would it do the same for MVA? And our first experiments actually was showing no, it was not. If we use the same way, the same method with these cells, we were losing productivity per cell when we used this. And we had to do something on what we were doing with the perfusion, so with the exchange of the medium over time in that virus production phase. Please, next. But finally, we were able also to use this ATF system, and we were using 2 different cutoffs of the hollow fiber and a specific perfusion rate that we were using. Next, please. And again, we could show that even the cell-specific productivity per cell, we were able to increase even with this strategy. So making even more viruses per cell by doing this. Next, and if you put this into numbers, we were able to get 14-fold higher titers, 2-fold higher volumetric productivity, and 2.5 higher cell-specific yield. And this at 50 million cells per ml. So in a small bioreactor, you can now go for 5-fold more cells, and with that, also much more productivity. And by just plugging outside a hollow fiber. So if you think of the capacity of vaccine production today, you just plug outside a perfusion system and you can already increase your productivity. Okay, next please. Well, then came, well, now we showed influenza, we showed MVA was possible, so what would be a next challenge? And then we came up with yellow fever and Zika. At that time it was very discussed a lot. And as I showed you, this is a virus that produces, if it's doing well, 10 virions per cell. So it really makes a change if you have more cells, if you're able to produce really a lot of cells. So this was really, okay, where can we go? And we had a really smart PhD student who worked on that. He first looked at yellow fever, then we were able to send him to Brazil, to Rio, to do first experiments with Zika there. And then he came back, brought the Zika virus into our labs, continued working on that, and improved and improved and improved. And what you see here is the outcome, the final outcome of what he was able to do. Next, please. So compared to the, he was even able with these perfusion systems to, with different strategies of the perfusion, to increase and increase. And at the end, he was reaching 160 million cells per ml. And he was producing and was even, again, increasing the cell-specific productivity. If you take this for yellow fever virus, what we were able to produce in a 1 liter bioreactor was really already a lot of doses of vaccine that we were able to produce there. Okay, so we thought, okay, we know these membranes systems, perfusion systems, that's fine, but what about the other perfusion systems? So whatever we were in these membrane-based systems, we always had virus also attaching to the membranes, and we lost our viruses. So not so good. So of course, interesting, can we use the other perfusion systems as well? And we were able to have this inclined settler in a collaboration with the University of Bielefeld. and what you see here, so how this is fit, and here you actually see it with the bioreactor. Next, please. And we were able there also to grow 50 million cells per ml and to produce even with higher productivity than in the batch or in these ATF systems that we were also looking at. So we could again increase the productivity per cell. Next, please. Yeah, further on we tried the acoustic filter. This is how it looks. So it's actually here in the very disguise, and this is how it looks, the compartment where you put the liquid. You can see here, this is yellow liquid, half filling half of the flow chamber. Next, please. And again, we were able to show that we could grow to 50 million cells per ml. Next, please. And for influenza virus, we were able also there to keep the cell-specific productivity, and we also tested it for MVA, and again, very promising figure, catching up what we were also able to do in ATF system. Next, please. I promised you that we also looked at the other way of intensifying this process. So what do you do if you prolong the virus production? And this is, you need to go into two-stage bioreactor systems. So you need one bioreactor that produces your cells and one where you have your virus infection, and you continuously feed virus into your bioreactor, into your virus bioreactor. So as the cells are dying with the virus replication, you add new cells. Good. So what you would expect is you have your cell that infects, you have your virus that infects one cell, and a lot of new good virus particles are produced. However, this is not the case. Next. And and if you have that, you would have expected that this would be the outcome of our experiment, so the virus titer in a continuous way produced. Next. However, with influenza virus, as it is an RNA virus, it also makes a lot of, well, non-infectious virus particles. And some of them, they have an interfering attitude. And if they are co-infecting with a good virus particle, what they actually do is they push down the production of the good virus particle, and is producing a lot of defective particles. And this made, next please, that our experiment didn't give this straight line, but it gave oscillations of the virus titers. And if you can also analyze these defective particles with PCR, and we could nicely see that really these oscillations were due to these defective particles that were out-diluted because of this continuous mode. So for influenza, this is not really an answer to produce in a continuous mode, because you lose your overall virus titer due to these oscillations. But for MVA, it's a different virus. It's a DNA virus. Next, please. We were able to show that it was working. I'm not going to show you due to the time. Sorry, but well, again, a smart PhD student had a good idea of what could we do to overcome this. And this is the answer. It's a tubular plug flow bioreactor. Next. So what it does, you have a tube that is moving through, where the virus and the cells are moving through the tube very slowly, and you have volume units that are separated by little air bubbles. And this is a 100-meter tube, and this gives you the residence time of the virus going through that tube that is long enough so that the virus replicates. But the viruses that are released are not infecting new cells. It's only the cells that are in that little volume particle, and you do not get co-infections in that system, so you do not get defective particles. Next, please. So compared to this two-stage bioreactor, we could show that we really get this continuous production of influenza virus with not these large oscillations. And on top of that, we have a defined virus passage number. Next, please. Yeah, and with that, I'm already at the end of my presentation. What I wanted to show to you is that we are actively working on showing a new platform of producing viruses with different modes. I didn't show all of them, but different bioreactors combined with different perfusion systems to go into high cell density, different ways of producing them in continuous mode, and already different viruses. We are looking into new viruses, of course, with new cell lines, and are, of course, also, again, trying to get the platform even larger. Next, please. So overall, we were able to grow the different cell lines already now to 60 million, 83 million, 160 million cells per ml, and producing these viruses to titers that, well, many people would love to produce, all of them above 10 to 10 infectious units per milliliter, which is really a lot. With that, I'm going to finish in acknowledging all the people that are involved. Next, please. So this is my team, the upstream processing team within the Bioprocess engineering group of Professor Reichel. And we have a lot of collaborations. As I said, we are allowed to do applied sciences, and with that, we do have a lot of collaborations with industry, and we're very grateful for that. And with that, I'm at the end, and thank you for your attention.